There are several categories of neurotransmitter. The first is acetylcholine. It's in a category all by itself. Then we have the amino acid neurotransmitters. This includes glutamate, the excitatory amino acid, and GABA and glycine, inhibitory amino acid neurotransmitters. GABA is the primary inhibitory neurotransmitter in the brain, while glycine is the inhibitory neurotransmitter used in the spinal cord. Neuropeptides are another category of neurotransmitter. Peptide just means small proteins. So these neuropeptides are a chain of as little as 2 or 3 or 4 amino acids that make up the signaling molecule. We've already talked about one neuropeptide when we talked about psychopharmacology, and that was beta-endorphin. It is, of course, the endogenous ligand for the mu-opioid receptor. The next category of neurotransmitter is a very large category, and it has 2 subcategories. We aren't going to talk about all of the monoamines, but we are going to cover at least one indolamine monoamine, which is serotonin, also known as 5-HT. We're also going to talk about 2 catecholamine monoamines, dopamine and norepinephrine, which is also known as noradrenaline. As a reminder, synaptic transmission includes a number of different steps. Some of them happen kind of behind the scenes, while others are directly related to synaptic activity or to activity of the presynaptic cell. Neurotransmitter production and packaging into vesicles happens kind of behind the scenes. Vesicle docking is also not related to whether or not the cell is firing an action potential. Then neurotransmitter release is triggered by the action potential. Once neurotransmitter has been released, it can bind to and activate postsynaptic receptors, which may be ion channels in the case of ionotropic receptors, or could be G protein coupled receptors in the case of metabotropic receptors. It's important to eliminate neurotransmitter from the cleft to make sure that the message the postsynaptic cell receives is still relevant when it's receiving it, and also to prevent neurotoxicity or excitotoxicity. In general, there are 2 methods for getting rid of neurotransmitter. The first is enzymatic destruction, and the second is reuptake. We'll talk about each of these steps one at a time for each of the neurotransmitters. First, neurotransmitter production. Amino acid neurotransmitters begin with the Krebs cycle. Glutamate is produced as a product of the Krebs cycle, and then glutamate can be further modified to create GABA. From glutamate, the enzyme glutamate decarboxylase, or GAD, produces GABA. Only inhibitory cells that contain the enzyme GAD will produce GABA. If the neuron is an excitatory neuron, it will not produce the enzyme GAD, and it will have only glutamate and not GABA. Synthesis of acetylcholine depends on the enzyme choline acetyltransferase, which converts acetyl-coenzyme A and choline into our small molecule neurotransmitter acetylcholine and a side product of coenzyme A. In the monoamines, we can think about catecholamine and indolamine synthesis as 2 related but different processes. Catecholamine synthesis begins with the precursor L-tyrosine. Tyrosine hydroxylase then converts L-tyrosine into L-dopa. Dopa decarboxylase converts L-dopa into our small molecule monoamine dopamine. Some cells will ultimately convert dopamine into norepinephrine. Those cells contain the enzyme dopamine beta hydroxylase inside the neurotransmitter vesicles. Dopamine will be pumped into the vesicle, and then inside the vesicle it will be converted into norepinephrine by the enzyme dopamine beta hydroxylase. only neurons that will produce norepinephrine make this enzyme. Neurons that are destined to be dopaminergic neurons will not produce the enzyme DBH, dopamine beta hydroxylase. Indoleamine synthesis is very similar to catecholamine synthesis, but the starting material is tryptophan, not tyrosine. Tryptophan can then be converted into 5-HTP using the enzyme tryptophan hydroxylase. 5-HTP is converted into 5-HT, aka serotonin, using the enzyme 5-HTP decarboxylase. Most of the small molecule neurotransmitters are produced in the terminal, the exception being norepinephrine, which is produced actually inside the vesicle. The neuropeptide neurotransmitters are produced via transcription and translation and then brought to the terminal inside the vesicle already. In order to package neurotransmitter into vesicles, we use a transporter protein. This transporter will be specific for the neurotransmitter in that cell, and it will use ATP to pump the neurotransmitter into the vesicle. Glutamate is packaged by the transporter V-GLUT, vesicular glutamate transporter. If you had a drug that blocked vesicular glutamate transporter, you would not have glutamate inside the vesicle, and therefore you would decrease synaptic transmission between the two neurons. That decrease means that this drug would be an antagonist. Because the drug is not directly blocking the postsynaptic receptor, this would be considered an indirect antagonist for glutamate. GABA is packaged by vesicular GABA transporter, or VGAT. It should not be surprising at this point that acetylcholine neurons have a transporter named vesicular acetylcholine transporter, which is used to pump acetylcholine into the vesicles in those cells. For monoamines, it's a little bit of a different story. Here we have vesicular monoamine transporter. This transporter is found in serotonin cells, dopamine cells, and norepinephrine cells. In serotonin cells, serotonin is pumped into the vesicle by VMAT. In dopamine cells, dopamine is pumped into the vesicles by VMAT. In norepinephrine cells, dopamine is pumped into the vesicles by VMAT and then converted into norepinephrine by the enzyme DBH, as shown in this figure here.